Hi everyone, my name is Chak Li, or you may call me Richard, who is going to present our work on integrating multimodal data through interpretable heterogeneous ensembles. First, I would like to talk about the heterogeneous ensemble, where the motivation is mainly because we are struggling on choosing the ideal algorithm to our target problem. And heterogeneous ensemble can aggregate different type of models into ensemble, and like by taking the mean of the prediction or using ensemble selection method or using stacking. And we have successfully applied heterogeneous ensemble on predicting protein function already, but which is only on one type of dataset. However, multimodal data is getting more and more popular right now, especially in biomedical research, which contain lots of information, which can capture the complementary aspect of a biomedical entity, such as like protein. We may have the amino acid sequence or the 3D structure, and also the experimental data of a protein. Another example is the electronic health records, or we call EHR, of a patient, which may contain the basic demographic of a patient, or also the laboratory test results from the patient blood sample, or the medical images and medication record of the patient, which all can help to make a better clinical diagnosis. However, how can we integrate these different data to predict on our target problems, and how can we interpret the predictive model based on the multimodal data? And the major integration approach right now is based on early integration, which cannot totally solve these two problems. And that's why we propose a systematic late integration approach on the multimodal data, named as Ensemble Integration, or called EI, which is built based on the heterogeneous ensemble. And for example, a multimodal data can be divided into different modalities, like the co-expression of a protein or the demographics of EHR data. And each modality can be build their own local models and capture the complementarity. And finally, these local models can be aggregated by the heterogeneous ensemble algorithm, like I mentioned before, like taking a mean or using stacking of it to build the EI models. And with the EI models, we will first assess the prediction performance and then perform the interpretation on the best performing ensemble, which allow us to find out the key features to the target problem and also build up the trust to the user. And we have tested EI on two representative biomedical problems, one on protein function prediction, the other one on COVID-19 mortality prediction. In the protein function prediction, we use the string data to predict the annotation of gene ontology terms. And we use the EHR data from the patient treated at Mount Sinai to predict the COVID-19 mortality. And this model can help the hospital to provide a better healthcare to the patient, especially the serious one. And then these two experiments were evaluated based on a nested cross-validation, and the performance were measured in FMS. We compared EI to other integration approach tools such as Mashup and DeepMF, which were the early integration representative for biomedical networks. The results shown here were based on the best predictor trained on the embeddings, and the others shown here were the individual modality of string, which is using heterogeneous ensemble modeling it. And we find that EI is performing better than mashup and DeepMF. The possible reason behind is because EI is fully optimized to the target prediction problem, while mashup and DeepMF first build the universal embedding for not only the prediction but other analysis. And we also compare EI to other models on the prediction of COVID-19 mortality, where we first find out that EI is performing better than the other individual modalities. Where I would like to highlight that lab test here is the best performing individual modality, which I will talk more about it in the model interpretation part. And we also find that there were two EI models is able to perform better than the XGBoost, which is a other early integration approach by the first concatenating the EHR data and then train based on the XGBoost algorithm. Here, I would like to talk about how can we interpret the EI model. Since EI is built based on heterogeneous ensemble, which contain two layers of prediction, we need to do the same to the interpretation too, which first getting the local model ranks and also getting the local feature ranks. Combining these two together, we can assess how important the feature contributing to the EI model, and the combined score we name as rank product score were sorted, and then we can finally have the feature ranks of the EI model. And here is the key feature found to be predictive to COVID-19 mortality, and we found that there are some consistency to other results. First is on the prediction performance, where the lab test is the dominant modality of the key features, and also some of the features here is overlapped to the features found from the XGBoost model, and it's also the most important thing here is it's also consistent to the existing clinical study on COVID-19 mortality, where like the age, the minimal oxygen saturation, and the respiratory rate were the key feature to estimate the risk of COVID-19 mortality. To conclude, EI is able to enhance the prediction performance on the biomedical problems, and the interpretation of EI models can identify the key features of the target problems too. In the future, EI can be expanded to other multimodal data, especially the unstructured one, and there can be some further improvements on the prediction and interpretation algorithms too. I would like to first thank the author of this work from either Pandy Lab or Virginia Tech, and also other colleagues in Pandy Lab and Mount Sinai too. And the computational experiments were done on HPC of Mount Sinai and Oracle too. And finally, I would like to thank the grants provided by NIH. And also, this work will be presented in the poster session to F007, so we will see you there, and thanks for the attention of this talk.